Looks like. Okay, welcome everyone. Before starting, I really want to acknowledge, I can say, the organizers of the Novacon conference and all the officials of West Bengal. So I learned a lot from the poultry industry here. It looks really impressive. And I really like to be a small contributor of your innovation conference. Innovation is really what really is needed to become leaders in our world. It was quoted before. So that will be my milestone, to say something like my contribution here to help you better understand how vaccination works and what you can expect from vaccines. And in the title, there is something related to what we call serology. That may be interesting. We are all here scientists, maybe not really working in that field. And serology is a way really to monitor vaccination, to monitor disease prevalence. So that way, again, we have to link the two. I mean, vaccination development, how to bring innovation in vaccines, and what will be the consequences on serology. So that means, like I said, the tool that you may use for disease prevalence or vaccine take. So this is overall the topic of today. Are you ready? Okay, good. Let me move. The slide doesn't move forward. I need some help. Please yes. Before we start, I think this topic in this presentation today, can you help me? That doesn't really move forward. I'm going to explain you what are the different platforms, what we call platforms, the different technologies to develop vaccines. And we'll review everything related to the monitoring. But before we get started, so you have to hint, it's a very, very short view on my next slide. We'll explain you a little bit on what is the vaccination strategy. What is vaccination? So let's wait for the next slide. I'll let you think about it. Vaccination. We are talking about microorganisms. So you know we are talking about poultry, vertebrates, and birds. But here, this is a different world. We are here in a microorganism world. We're dealing with viruses, with bacteria, and mostly, we may even fungi. All these tiny organisms are really causing disease, as you know. And we need vaccines. It's obvious. It has been really more than half a century we've been developing vaccines. It has been really becoming one of the, what they even call sometimes a commodity, or at least now, what we can consider as well, because the technology moves forward so fast, that is also innovation brought to the farms. And the vaccine will prepare, as quoted here, the immune system, I don't know if it shows, okay, the immune system here for contact with real pathogens. Ideally without causing harm. So the vaccine is here to stimulate the immunity, the immune system. And it is what we show in the bottom slide of this slide. We've got here what we call the stimulation of the immune system. The immune system of the bird sounds a little bit similar to the human immune system. There are slight differences. But overall, I think it works more or less the same way. We've got here very important cells called white blood cells, and they are here coded as B or T cells. They are what we call lymphocytes. And these mechanisms, without giving you all the details of the immunology, immunology is a science that is dealing with all these mechanisms here. What is really important to take away, to understand, is that your tiny green things with what we call antigen presentation, all the proteins that are above the surface of these viruses, these bacteria, will help produce antibodies, for example, or what we call an immune response. And when you use vaccines, the idea is to stimulate this immunity and those pathways. So this is the overall strategy. Important here, I can say among us, really to differentiate what is mucosal and systemic immunity. It's a lot of definition here. I make it really simple, catchy. Systemic immunity, pretty easy to monitor. Serologic antibodies, you know you do, and we do in chickens as well. Maybe some of you are not really aware of that one, but we do serologies. We take blood and we go, let's say process these blood samples in the lab. And we've got antibody profiles against various diseases. You've heard about Newcastle disease, you heard about Gumboro disease, you've heard about Marex. Marex is not a good example because the antibodies are not really there, or not detectable by the current technology. We've got here, though, this is what we call systemic immunity. We can really monitor it out of the blood. So that means that it will protect the whole, viremia, the whole multiplication, replication of the virus, for example, in the body. And what the mucosa, we all understand what is a mucosa. This is a local layer, of cells. It could be the respiratory mucosa, it could be enteric mucosa, everything that is in contact with the outside environment. And this mucosa is pretty important because it's a kind of barrier. So you can say it's a border between, the body, the inside, and the outside. What is typical from the mucosa is the respiratory. It could be trachea, it could be our lungs, and so on. So this is where we have another type of immunity that is called mucosal. Your vaccination will help stimulate the immunity, and you've got mostly two main types of immunity: systemic, the blood, and the mucosa. What are the main vaccine types? You've got the kill vaccines, you've got the live vaccines, easy. And we've got a little bit more complicated, let's say the new technology. It could be the subunit. That means that you don't have the full pathogen, the whole virus, for example, but you take only one single piece. That will stimulate the immunity. That will be enough. You have the vector vaccine. That's a different approach. You've got a carrier. You've got here a safe virus, for example. You just insert the gene that will code for another antigen, another protein from another virus and stimulate against two diseases. But we'll review it one by one. When we work with live vaccines, in fact, all these live vaccines, so this is the first platform. We'll check one by one. So live, kill, remember, subunit, and vector. Coming back to the live vaccines, these live vaccines are what we call attenuated. That means you don't take a plain E. coli, for example, and just inject as it is. No way. You need to attenuate. Same with the viruses. You take Gumboro virus, IBD virus, you remember, will induce immunosuppression, bursa lesion. This is this tiny organ that is related to the immunity. But if the virus is not attenuated enough, it will be inducing lesions, and even sometimes it could be a disease by itself. So attenuation is pretty important. This is what we see here. And say on this diagram, we can have, for example, serial passages. What good example? H120. I'm pretty sure you've heard about that one. Infectious bronchitis live vaccine. H120, for example. 120 is about the number of what we call here serial passage of the virus. It's a lot of work. And how to monitor vaccine take. Remember, a live vaccine will replicate mostly in the mucosa. Infection localistis is a very good one. It will replicate mostly in the respiratory tract. Not only, but mostly. And this is interesting. Newcastle disease live vaccine will be the same. And what we show here, because we've got different types of monitoring here. We can monitor, and this is what we see on the right-hand side of the screen. This is interesting to see. This is the serological monitoring, the blood, systemic immunity, because any type of vaccine will stimulate the systemic immunity. You can monitor with the blood. But maybe not enough. We also can monitor with what we call the mucosal immunity. This is not very common methodology we may use in India, even worldwide. But this is more on the science side to explain, to drag your attention. What is really important with the live vaccines, it may be not what you really monitor. We all like to monitor the blood antibodies. Of course, it is much easier. But we may also show you that we have some, and this is what we see, on the left-hand side. We have some of the monitoring using different methods. Two of them are listed here, because the HI test, inhibition of hemagglutination, and ELISA. And these are here local mucosal antibodies. And what is really important to tell, I wanted to conclude on that one, is that the two segments of immunity, systemic and mucosal, are linked together. That was explained in my former slide, and this is really important for you to know. I mean that this is not disconnected. I mean once, you've got some immunity, local mucosal, I mean you may have systemic immunity as well, but not as sustained and not as high as with other platforms, especially here the killed vaccine. The killed vaccines are totally different. It's a different approach. So you remember, we go one by one here with the different, what we call, technology platforms for vaccine development. So I was speaking about the live vaccines. Now we call on the next one, that is the kill vaccine technology. What do we do? And this is not specific to poultry. I mean, we've got human vaccines as well. These kill vaccines usually use the full microorganism, the full virus. It could be avian influenza virus, not pretty common here in India, other countries. It could be the Newcastle, more popular virus known here within the poultry industry. It could be the IBD, infection localistis. Infection localistis, it's a big one in India, for sure. And all these microorganisms, as we can see, they have a step of what we call inactivation. So this is what we see here. I mean, we show here more bacteria, but same with the virus. We inactivate them, we kill them. That means that we have the whole virus or the whole bacteria shown to the immune system, but usually it doesn't work on its own. You need what we call an adjuvant. So this is what is shown here, adjuvant. And this is another part of it. This is really part of what I call basic vaccinology. When you have a kill vaccine, what we call an antigen, so you can read it and some of the formula of the vaccines. What is called antigen, like I said, this is really this part that is prepared for immune stimulation. You got the adjuvant, that is an additional content that is not intended to stimulate the immunity specifically, but it will help stimulate the immunity. And this adjuvant is very important. And for poultry, and some of you have seen these inactivated vaccines, most of the time it looks like it's an oil emulsion. And this is really specific to poultry. If you are familiar with other mammal species, it's not that common to use those oil emulsions, but the birds are taking it pretty well, and it stimulates very strongly the immune system. And how? I still have a problem with the, no, it's all right. Sorry about that. Okay, so that will be totally different. Remember what I was telling about the live vaccines. So we've got mostly mucosal local immunity, antibodies more locally, somehow, some systemic immunity, but not really long-term and sustained. The inactivated kill vaccine, that's the same, inactivated or killed, will, total opposite, induce a long, sustained seroconversion. And this is interesting, and systemic immunity. This is exactly what we see here. These are really simplified diagrams. And you can say above my above curve, that is a blue one, it's just what we call prime boost protocol. We've got the live vaccine, and we've got here this Newcastle disease, pretty common, because everybody is vaccinating against Newcastle in India. And we see that when we have a kill vaccine, as compared to the below curve, that is a red one is only live vaccines. When you got a blue above curve, this is here the, live plus kill vaccine. So this is really a way to have a sustained, continuous, high-level immunity that will protect here, especially against virus circulation, contact with the Newcastle disease, velogenic one, and in layers, if some of you, have laying birds, they will also protect against egg drops induced by Newcastle disease virus. And meanwhile, this is what we can see, in the below graph, and meanwhile, we correlate this sustained antibody response with protection against challenge. So it was lab challenge here, using a velogenic Newcastle strain. Subunit vaccines, little by little, are making their way to India. I'm not sure that we have plain registration yet for such vaccines, but this is important to anticipate, and this is Innovation Conference. So we have different ways now to better target what is really needed in a microorganism, to simplify the process of production, for example, and even target the immune system, if I can say a more better target of the immune system against really a proper protein of a virus or a bacterium, for example. And this is what we call subunit vaccine. So that means, I take an example that we all talk about it, even if we don't vaccinate here in India. Even influenza, it's a very good example, and it's really, you can say, explaining that type of technology. You have different antigens on the surface of the virus. One is called hemagglutinin, the H5. Everybody knows about the H5. You've heard about it. This H5 is the main protein, the main antigen, as we call, that we use here, what we call expression system, a system here to produce this H5 antigen and formulate it with an adjuvant to produce what we call a subunit vaccine. It will not be full virus, but will be targeted antigen. And the main advantage, if you do the monitoring by serology, is that you can differentiate the infected birds from the vaccinated birds. The vaccinated birds will show antibodies against H5. The infected birds, infected or vaccinated or not vaccinated, all positive for H5. But we can use another protein for the monitoring, it's for example the NP, and if you have infected birds, vaccinated or not vaccinated, it will be NP positive. If it's vaccinated and infected, you can say, it will be H5 and NP. If it's vaccinated, if it's non-vaccinated and infected, it will be NP. So there is a way really to differentiate infected, vaccinated. The vector vaccine are very interesting, and I would say really, I can say this is the today technology. Obviously, this technology is not new. I started to work, I can say, last century, more than 30 years ago. It was, you can say, we were just starting with these technologies. Today is really, it's a given. You have it on the market, and in India. And this technology is pretty interesting and straightforward. You take a bacterium, a pathogen, a virus, an HVT, for example, that you can use in a hatchery to vaccinate against Marex, and you just insert a gene that will coding for another pathogen. That could be, for example, a IBD virus, protein, and again, such as it could be the H5 or even influenza, for example. It's a busy slide, but what is really important to tell is that the vector vaccine technology is quite interesting as long as you have a lot of room, very big DNA, to make that insertion of another gene workable. And this is again the reason why I could say this is really again a unique platform. And each vector technology will have its own platform, own characteristics, and will enhance immunity because of the in vivo replication pattern. What is interesting, coming back to what I was presenting about the subunit vaccines, there will be the same principle. You've got an HVT, you've got an HVT plus IBD, another virus, so two. You're not going to monitor with serology the HVT. That's not working. But you can monitor the IBD. And you can monitor the IBD VP2. That means really, again, the inserted antigen, the inserted gene. And this is exactly what we see here, seroconversion against VP2. That is really specific. And if you use an IBD monitoring system, you are not going really to monitor specifically the vaccine take. It's kind of a semi-DIVA system, differentiating infected and non-infected animal within the vaccinated birds. Interesting, same story with the HVT and the Newcastle insert. It could be single or double insert. You've got the vector vaccine now with one single insert, but some of them with two inserts. And we're already working on 3 inserts. So that's the technology ongoing. What is interesting with Newcastle is that we have another antigen that is inserted that is coding for what we call the fusion protein of the Newcastle disease. And this one you can monitor by serology. And this is specific serology because these antibodies will be detected only with a vector vaccine. And this is very interesting because you can somehow, this is not a strict DIVA system, but it somehow gives you some insight on vaccine take using that specific platform that is here detecting fusion protein, that is really the insert of the vector vaccine. And you can monitor meanwhile using another test. This is what we can see with the NP, same principle as the avian influenza. So you can detect the whole virus. So it could be an infected field infection, for example, but it also could be a live vaccine. Interesting, even not present here in India, but pretty interesting more for the science background. We launched that vaccine in different geographies, especially across Asia. And what is interesting with the laryngotracheitis, this is an herpes virus with different glycoproteins outside. And I would say each vaccine, vector vaccine, has a different insert from different glycoprotein. And what is interesting with this specific virus is that you can monitor strictly, let's say, the vector vaccine with the proper insert. So the proper insert here, for example, in the example I'm providing, is the GD, is one of these, as you can see, glycoproteins. And all these Gs, as you can see on my slide, are, again, more than 12 of them. And this is what we call the ultimate way to say, and we try hard, again, to deliver these kind of technologies with our partners, because here we're working as vaccine producers, developers, and in diagnostic lab kit producers. Together, really, this is a teamwork. And the teamwork is really to help define the genetic markers of these vector vaccines to bring specific monitoring systems to help monitor vaccine take. This is really a breakthrough. When you work with live or kill vaccines, there's no way that you really do that. And this is what we see here, for example, with the ELISA based on GD, so this insert or glycoprotein specific to the vector vaccine. So, I'm almost at the end of this presentation, so I hope you can say you had some understanding of what we try to do, linking vaccine technologies, vaccine development, and what is really required from the field use, and especially what can we do with the tools we have. I mean, the tools we have are diagnostic kits we use in labs for monitoring. We see here that when we have the in vivo presentation of the antigen, we've got here the full inactivated vaccine. It could be, and not very familiar yet here in India with this technology, the reverse genetics. Very short word on that one. Reverse genetics is a way, remember, to take a whole virus and just, even influenza, for example, and just swap some of the genes. It's kind of what I call chimera type. Overall, you can have a full virus presentation, but the backbone of the virus will be a mild, can say low pathogenic one. So this is really a trick to allow a full virus presentation. And this one, again, we see seroconversion linked to contact with the presented antigen. And these antibodies will be detectable if no disease challenge and imperfectly detected with disease challenge. The big challenge we have with the full virus is that it's very difficult, as worded here, to make the difference between your vaccine take and your infection. And maybe some of you are veterinarians, poultry vets, so they are probably familiar with the art of interpreting the serologies. Most of you rely on what those guys are doing. And overall, believe me, it's not that easy. You need to work with baselines and have a very, very long experience of interpreting those results. The subunit vaccines can say that's really a big advantage because it simplifies the overall interpretation of your serologies. It's a little bit complicated, a little bit because you may use two types of ELISA kits, for example, but overall, you may differentiate. Again, that's pretty interesting. I can say the vaccinated birds infected or not infected. Remember, if they are infected, you use another test to say this is a full virus presentation. If it's only vaccinated, it will remain. Remember my example with the H5 positive in any case, but no NP, no other test positive. And for the in vivo expression of the antigens, this is interesting here. Remember what I was showing before? The in vivo presentation of the antigens. You just inject, and the immune system is just processing what you inject. So this mostly kill vaccines, subunit vaccines. Everything killed. When we speak about in vivo expression of the antigen, we talked about the viral vectors. Remember? By HVT, for example, it could be another virus, could be fowlpox, for example. There are different platforms. And when we say in vivo expression, they mean that these viruses will replicate, will multiply in the birds. So the poultry, vector vaccines, are strong replicating viruses. So they are there to replicate and to be a little bit of everyday vaccinators inside the bird. So that's really a very, very powerful way to vaccinate. And it can be used in a hatchery. So that's very interesting because that's really the way, I can say, we've got a breakthrough technology here. And the monitoring is specific because it's just like the subunit, same principle. You got here one single antigen you can monitor. And I'm giving here, I can say for the very last comment here, I can say some, I would say, tips of what is happening now, I can say among all the technologies. This is becoming wild. We'll have a lot of different platforms. Remember all the COVID. We all suffered from COVID. Remember the RNA vaccines, humans vaccinated with them. And the poultry vaccine technologies, we try to learn, to catch up on everything that was already on the market, especially for humans. So RNA vaccines is one of the approaches for the future. And that will be my conclusive remark here for this Novacon conference. So again, I'm really a fan of your initiative. I can say to focus so much on innovation. This is really the future of the poultry industry. And with that, I just want to thank you for your attention.